Okay, welcome everybody to session 6, AI in drug discovery. Artificial intelligence, as we know, is a very interesting technique and allows us to infer new meaning from complex data sets. It's used in a variety of applications, but perhaps the most promising of which is drug discovery, and we're going to hear about that today. So we're going to start off with Fergus Imrie, a fellow at Oxford, to talk to us about OpenBind.
Great. Thank you very much for the introduction. So this is my first CCP4, and I've certainly learned a lot in the time here. So thank you very much for the invitation. And I'm delighted to be here speaking to you about some of my work and the work of the OpenBind consortium more generally, and how we can use kind of data in machine learning to hopefully unlock protein-ligand binding prediction, which I think is a very nice complement to a lot of the work being done in actually the data collection side of things as well. So over the past 7 years, I think we can probably all agree that we've seen this huge revolution in computational structural biology, primarily driven by AlphaFold, which was released in 2018, and then AlphaFold 2 a couple of years later, which was then awarded the Nobel Prize in 2024. So I'm assuming everyone probably knows what AlphaFold is, but as a 10-second primer, AlphaFold's a deep learning-based model that predicts the 3D structure of molecules, such as proteins, from an amino acid sequence primarily. And so I think now that we'll pretty much agree that we have the ability to relatively accurately predict at least single domain proteins, even in novel folds from their amino acid sequence alone. And there have been many extensions of this methodology, trying to extend it to new macromolecules or even protein-ligand complexes that have been proposed in the last couple of years. So as I mentioned, one such extension is to protein ligand complexes. And I think as others have mentioned, the ability to predict these is almost one of kind of the holy grails, I think as Phil called it in his talk, of computational drug discovery. So can we predict not only the co-complex structure of the protein and ligand directly from, let's say, the amino acid sequence and just the molecular structure, instead of using kind of a two-step modeling approach, or needing an experimental structure is necessary in kind of docking. So AlphaFold 3 really was the first method to do this. It was released kind of about a year and a half ago now. It was quickly followed by several other approaches, such as kind of Boltz, Chai, NeuralPlexer, and most recently, OpenFold 3. So on this slide, I'm showing an animation of some of the outputs of the recent Boltz 2 model. And this method claims to not only be able to predict the structure of the protein ligand complex, but also to relatively accurately predict their binding affinity between the two. And I think I say claims because I think we've already heard in this meeting a few disputes to this claim, and this is something I'm going to speak about a little bit more throughout my talk. So all these claims sound great, but I want to start by just talking about how good are these methods really? Are they doing kind of what they say they are? So the first thing to note, I think, that's been established in the last year is the performance really is highly dependent on training set similarity. So on the left, I'm showing one of the figures from the recent runs and poses benchmark, where they really looked at the success rate of these methods, and I won't define success rate too much here, but kind of just, is the structure good enough by some measure, measured against the similarity of that example to things the method's seen before, and that's both sequence similarity as well as kind of ligand shape similarity in the binding site. And as you can see, all of the kind of co-folding methods that were tested have this really strong dependency on how similar is this to something we've seen before. And this might really not come as a surprise, and we can argue about whether or not success rates are in the kind of teens to low 20s for these complexes that look nothing like we've seen before is good or not. But I think this is one thing that really shows these methods haven't learned some kind of generalizable representation or ability to predict protein-ligand co-structures. On the right-hand side, some really recent analysis from Eva Nittinger and colleagues at AstraZeneca that looked at the differences in these methods in being able to predict allosteric binders and orthosteric binders. So the ligand RMSD is shown here on the x-axis, where kind of as far to the left as possible is good for a variety of different targets. And in blue are all the orthosteric binders. And you can see the majority of these methods put in kind of the right place. And these methods have seen lots of orthosteric binders. So again, as the figure on the left might suggest, this might not come as much of a surprise. And in contrast, it very rarely puts these allosteric binders in the right site, primarily just because we haven't seen that many in the data that these methods have been trained on. So there's this really, another example, there's this really high dependence on the training set similarity for these methods. I think additionally, we've seen that current approaches generate physically and biologically implausible structures. So on the left, I'm showing kind of the headline results from the PoseBusters analysis that was done about 1.5 years ago now, that looked at assessing protein-ligand complexes that were suggested by various docking methods, as well as both classical and machine learning, looking really at their physical validity. So simple things like, is the protein clashing with the ligand in the predicted structure? And you can see for the classical methods, this is rarely, if ever, the case, whereas all of the deep learning methods have significant limitations from this perspective, and the majority of their kind of structures clash. So in the shaded bars is how many of the structures kind of look good according to the traditional, is my prediction within 2 angstroms of the experimentally determined model, whereas in the solid bar is if you include as well some very basic physical validity checks, what that success rate then becomes. And you can see a really stark drop-off for a large number of methods. More recently, on the right-hand side, this was extended by some people at, I guess, what was Exscientia, to look both also at interaction recovery. So are we recovering things like kind of hydrogen bonds, pi stacking interactions that were present in the ground truth structures? And again, you can see that these methods, in particular the machine learning methods, do a very poor job at proposing such interactions, with the co-folding methods the most affected by this. And finally, turning our attention to binding affinity prediction, here's a lot of data that was curated by Guy Durrant, a PhD student from my department that's just graduated, actually, looking at the performance of a variety of machine learning methods that were trained on the PDB-Bind dataset and tested on this CASF 2016 dataset, so this kind of standard setup that Phil spoke about in his talk a lot. And you can see that kind of since 2015, there's been very limited kind of, if any, progress, at least within this experimental setup, despite a lot of different methods being produced. So it kind of appears that maybe there isn't such progress in this area, despite there being a lot of kind of attention and work on method development. So for the next few minutes, I just want to start by talking briefly about some of the efforts that I've been looking at in order to improve these various factors for these methods. So this will revolve around both kind of data as well as methodology, and I'm going to start by discussing some of the more methodology-focused work. So starting with docking, as we said, we see these really big issues with physical plausibility, and that the generated poses really don't make the interactions we expect them to. So looking at all of kind of the machine learning methods, while still not great, diffdoc was by far the best performing method, at least for both of these papers. And just as a brief primer, diffdoc is a diffusion-based docking method So you start with kind of the protein and the ligand, and then you kind of run a diffusion process. I won't go into this in detail, where the factors that can change are the translation of the ligand, its rotation, as well as all the torsion angles. And once you've run this process, you end up with a set of candidate poses that you then rank and confidence score with some other machine learning model. And this forms kind of your candidate protein ligand poses. So one of the issues, I think, and a fundamental limitation with the diffusion objective kind of in this domain is that it doesn't really align with the downstream objectives that we often kind of care about, except when that diffusion kind of objective is in kind of the perfect case, when that loss would be zero. So if we consider these three structures, so in the middle is the kind of the experimental ground truth, and then I've got two slightly perturbed structures with the same RMSD to that crystal structure on the left and on the right. One of the challenges is kind of the left is kind of physically impossible. We can see there's this clear steric clash at the top of the structure, whereas the right is at least kind of plausible if you would argue that it's a little far away from making that potentially key interaction. But yet both are considered kind of equivalent under the diffusion objective. So we're telling our model when we're training it, yeah, either error is kind of equally good or bad, whereas I think we'd all agree that the structure on the left being physically impossible is clearly a lot worse. So I wanted to ask the question of how might we train machine learning models differently to take these kind of factors into account? So with a PhD student at Oxford, Henry, we wanted to ask the question is, can we train models such as DiffDoc to account for things like physical plausibility? And the method we looked to do so was reinforcement learning, because we're not constrained within a reinforcement learning to that specific diffusion objective, but we can tell the model kind of under any criteria, including non-differentiable ones, this is a good outcome or this is a bad outcome. So a little bit of kind of maths. I don't want to go through this in any detail, but the key is that we can reframe this denoising process of any diffusion model as kind of a multi-step Markov decision process, and then basically apply a lot of really standard machine learning techniques. So the question is, does this kind of all work? So we took one of these methods, in particular, we took DiffDoc Pocket, and we fine-tuned it a little bit with reinforcement learning, where we were rewarding the model for generating things that were not only within kind of the standard 2 angstrom RMSD cutoff, but also were valid according to the postbusters validity checks. And looking at this for kind of different sequence similarities to our training set, we can see that across all three buckets, the system that's been fine-tuned with reinforcement learning in green generates significantly more poses that are physically valid than the baseline kind of original version of this method. So this is just things that are valid, but does it also still produce kind of low RMSD poses? And the answer is, thankfully, yes, we do see kind of still an improvement in performance when we do this fine tuning. So on the left, so we typically generate not just one candidate pose, but a number. So we generated 40 for every single target. And on the left, we're showing the results for the top one ranked pose, so the one that was scored by the model the most highly. And on the right, we're just taking the best out of all the poses with some knowledge of the ground truth. And you can see kind of in both cases across all sequence identity bins, this reinforcement learning procedure seems to help obtain more poses that are not only kind of low RMSD, but also valid according to these kind of postbusters validity checks. We also took a little bit of a look at whether or not we're recovering more of the ground truth interactions when we do this, and found that this was indeed the case, although I'm not showing this here. Moving back kind of to binding affinity prediction, I think Phil very correctly pointed out that there are some fundamental flaws with this setup where, right, we're training on PDB binds and testing on this CASF test set, but this is kind of what everyone's using. So instead of asking kind of, are we getting better at this? I wanted to ask a slightly different question and say kind of, when we do this, what are these machine learning scoring functions actually learning? So to do this, I asked the question instead of do protein-ligand scoring methods understand common binding interactions? So we can argue about exactly how key making more interactions is for improving compounds, but I think we can kind of all agree that most binding is kind of at least governed by a lot of these kind of key intermolecular interactions. So where these are just based on some analysis from about 10 years ago now, the relative frequencies across structures in the PDB. where you can see most structures obviously have these kind of very basic hydrophobic contacts, but there's also a lot of hydrogen bonding, pi stacking, and so on. So do these machine learning methods understand these interactions? So to probe this, I used a technique called model behavioral testing. And so effectively, what this is you can have some black box system, you have some input to it, and if you make some change to that input, you then analyze what change that makes kind of in the output. So in a kind of a binding affinity prediction sense, one thing we could do is we could look to remove a hydrogen bond that we know is present in the ground truth structure and see what effect that has on the prediction. So in this case, if we were to mutate that threonine shown in kind of the cyan to a valine, what effect would that have on the prediction? But also as a control, you can see I've highlighted in kind of purple another threonine in the binding site that's close to the ligand but not making any kind of hydrogen bonding interaction. So if we were to mutate that, what would be the effect of that change? Kind of as an orthogonal control case. So here's that kind of example worked through where I'm just showing the scores from kind of a classical physics-based method, VINA, and the scores there. So it assigned the experimental structure a score of -7.45. When we removed that hydrogen bond, you lost about a kilocal of energy in its prediction. But for the control case, the prediction stayed essentially the same and actually increased just a tiny bit as there was a little bit more kind of hydrophobicity in the binding site. There's a little bit more kind of bulk and grease. So that's kind of the behavior we might expect and you might think might happen. So we can look at this not just for hydrogen bonds, we can look at this for pi stacking interactions as well as kind of salt bridges on the protein side by making simple mutations to residues. And we can also look at this on the ligand side by kind of removing hydrogen bonds on the ligand by changing the donor or acceptor group, just let's say a methyl, a carbon atom, as well as looking at what happens if we add clashes via methylation or indeed adding kind of strain. So these 6 different checks I wanted to investigate and do so over a variety of methods. So for kind of that classical baseline, just using the orthodox VINA scoring function, and then looking at a variety of kind of feature-based machine learning methods exemplified by RF score, and then a number of deep learning methods as well. So as a start, just how do kind of classical methods perform on these kind of tests? So for each of these 6 different modifications that I was wanting to look into, I'm kind of plotting 2 things here. So I'll walk through this one a little slowly, and then you'll kind of get the idea for all of the other charts. So let's just look at the far left, kind of the hydrogen bond on the protein modification. So on the left-hand side of that, we're seeing what happens in that intervention case. So the red line is if there was no change in affinity or predicted affinity, sorry. And anything kind of below that line shows that in that intervention case, the prediction was reduced, which in all of these cases is what we would expect to happen. And on the right-hand side is what happens in the control case. And so we would expect that in most cases to pretty much stay the same in general, and certainly to see a bigger effect in the intervention case than the control. So for the classical method orthodox VINA, for 5 of the 6 tests, we saw exactly the behavior we're expecting. With the only test we really didn't see this kind of clear behavior is this kind of internal strain of the ligand that isn't actually accounted for in the scoring function. So this kind of suggests hopefully that these examples are somewhat reasonable to use to assess whether or not these methods are taking this into account. So how do the various machine learning and deep learning methods do on these tests? So to start with RF score, and you can see essentially across all of the different tests, it makes absolutely no difference with any of these modifications on the method's predictions. We can ask the same question of various deep learning methods, and across all of them, I'm only going to show a couple of examples. Again, we see very limited differences. Although the deep learning methods kind of change their predictions more, it's very random, and on average, we see really no change at all. And this is the case across a number of different deep learning methods, all of which were kind of trained within this standard setup of training on PDB-bind, just in general to predict the binding affinity, typically from the crystal structure, but in some cases from kind of a redocked version of that structure. The only method across everything I tested that had kind of any behavior, even kind of resembling what we might hope to suggest that maybe this has learned something about binding interactions, was the NINA model from David Koz's lab at Pittsburgh, trained specifically on the crosstalk data set with a bunch of different training objectives, where we saw some ability to kind of differentiate between these kind of intervention and control cases. Although note that the magnitude on the y-axis in all of these cases is a lot smaller than for the kind of classical orthodox Wiener method. So this suggests, I think, in general, that most of these deep learning methods kind of aren't learning too much about true kind of these kind of protein-ligand interactions. But at least there's some hope that at least one method has seemed to learn something. And kind of currently I'm looking into investigating kind of why this is the case and whether or not we can train these methods to take this into account more and learn this kind of what kind of view of protein ligand complexes that incorporates these kind of known interactions. For the last kind of 5 minutes of the talk, I just want to turn towards the other efforts with OpenBind. So I've spoken about methods a lot so far, and now I really want to speak about data. And I think one of our kind of key hypotheses about why we're struggling with a lot of these issues is actually kind of limitations in just the quantity and quality of the data we've got available, as well as some slight methodological issues. With that, if we can fix both of them, we should hopefully have a lot more powerful and useful set of models. So the main resource that I mentioned a few times for this is PDB-Bind, where we have about 28,000 protein ligand structures that have been experimentally determined that also have kind of a paired affinity measurement. So we know not only the structure, but kind of how tightly these bind. In contrast, so the PDB, which obviously methods like AlphaFold were trained on, has about 250,000 structures, and so kind of an order of magnitude more. And if we look at where kind of 28,000 sits, kind of takes us back to the PDB in 2004, 2005 days, which I think for me, when I looked at that, was a pretty stark reminder of how much further behind we are on this type of data than not. And I think it's really one of the credits, one of the things that AlphaFold certainly credit is the availability of data in the PDB. So this really leads us to believe that this lack of data is a limiting factor. And it's not just kind of the quantity of data that is limited, but also there's a lot of kind of redundancy in that data. And the protein ligand structures we have really only cover a fraction of possible pocket space. So this was some really interesting analysis done a couple of years ago. Now, they looked at all of the structures in the PDB and tried to identify all pockets and potential pockets of those, both from kind of PDB structures as well as AlphaFold 2 predicted structures. So probably the graphs, kind of the figures to focus on are the two on the top right in red and orange. And so we're seeing on the left-hand side is all of the pockets for which we actually have ligands in the PDB, whereas on the right, it's all of the pockets that are predicted to exist more generally. And you can see the pockets for which we have ligands is a relatively small subset and very clustered in certain areas of that kind of overall pocket space. So OpenBind is this new initiative that's really hoping to address a lot of these challenges. So OpenBind's kind of a multi-institute consortia, primarily based at kind of Diamond Light Source, with close collaboration with the University of Oxford, as well as partners from the Washington Institute of Protein Design. You'll be hearing from Frank in the next talk, who's partnered with part of the OpenBind consortium as well as other collaborators from kind of Columbia and the OpenFold consortium as well, amongst others. So OpenBind more generally is kind of this big open science initiative aimed at accelerating drug discovery by generating much larger, fit for purpose, high quality experimental data sets of paired protein ligand structure and affinity measurements, where for each target, we aim to have a much richer array of binders and affinity measurements. So I think fundamentally, we kind of believe that a lot of this technology from kind of the machine learning computational side exists, but it's really being hamstrung the moment by the data available. And so OpenBind really exists to generate this data and really quantify the impact of more data on these kind of questions. So OpenBind kind of includes this kind of full end-to-end pipeline from kind of target and pocket identification, protein engineering, crystallization, and structure determination through multiple DMTA cycles, as well as kind of model development, building, and retraining. So over the next kind of 3 to 5 years, we're aiming to double or treble the number of paired protein-ligand structures with protein-ligand structures with paired affinity measurements in the PDB, and kind of ultimately increase this maybe by an order of magnitude in the next 5 to 10 years. I think we believe if we do this, our computational techniques will hopefully improve significantly enough to really aid their use in active drug discovery projects and make them a lot more useful and able to really guide a lot more experimental decisions. And so the goal here is not just to kind of collect more data for data's sake. I think we'll only be successful if the data we collect is actually useful for the models. And I think this is one of the key challenges we have now, that we're trying to figure out exactly the best way of doing this. So just a brief words on how we're going to achieve this. So probably our primary design strategy is to build on the kind of the fast forward fragment methodology that's been developed over the past few years at Diamond Light Source, with a lot of collaborations with Oxford. So building on kind of the XChem fragment screening, we'll look to identify initial fragment hits for new targets, then propose fragment merges using tools such as Fragment-Stein and the fragment network, elaborate these merges with kind of retrosynthesis-aware tools like Cinderella, and then use various scoring methods to then sample from that large set of possible merges. These compounds will then be either purchased or made with robotic synthesis before then being evaluated both with kind of biophysics techniques like the cryoptics to determine binding affinity, and then screened at XChem to determine whether or not they bind and their structure. So this fast forward fragment methodology has been kind of now battle tested over the last few years at Diamond with a lot of the ASAP antiviral consortia that many people here can speak to who are actually involved in those, which I must say I wasn't. But I think now there's a lot of confidence that this methodology can work, and we're hoping to improve it further as we proceed through OpenBind. So in addition to generating and releasing all of this data as part of OpenBind, we also intend to run various community challenges to kind of both critically assess and drive advances in these protein ligand prediction models, as well as retrain our own models on the data that we produce, as well as other public data, and provide models for the community. These kind of large co-folding models are incredibly expensive to train, and very few people have the compute kind of required to train them. And so I think a key part of OpenBind will be kind of democratizing access to the latest versions of these models trained on as much new data as possible. So just kind of to conclude, so OpenBind is really aiming to increase the amount of data available, and experiments for this first set of targets are actually already underway. But I think I want to stress that just the data side is probably still not quite enough, and we need to improve both the data and methodology. And I hope kind of the first half of my talk convinced you of that. And I think one thing in particular as we go forward is, and something I've struggled with in the last year a lot, is it's essential to really devise more effective ways for us to assess these protein ligand complex prediction methods and whether or not they're actually getting better in real time. So with that, I'd just like to thank kind of the amazing students and postdocs I get to work with, as well as all of my collaborators, with a particular thanks to Henry, who led a lot of the work I spoke about on reinforcement learning, as well as my various funders. So thanks for listening, and happy to take any questions you may have.
Thank you very much, Fergus. Do we have any questions? Yes. I'll just remind everybody to say your name and affiliation. Start your question.
Stephanie Vankovich, Vanderbilt University. So I love the reinforcement learning that you guys are doing on top of it. Just curious if you guys have played around with different reward functions and sort of what have you actually found has been effective in this?
No, thanks. That's a great question. So the primary reward function we were using was the final, was kind of the number of the Posebusters tests that were passed by the generated structure, which includes the test for is it below 2 angstroms RMSD. So that was the primary reward. We also looked into kind of giving kind of additional rewards if you were also making various interactions that were present in the ground truth. And so different rewards for did you make more than a quarter of them, more than half, more than 3/4. There wasn't a huge change in performance when we added in those kind of interaction-based rewards. I don't know if we just needed to train for a bit longer, kind of still finishing that off a bit. It slightly reduced the number of things that were kind of posed as valid, but did improve the number that had more than 50% interactions recovered. So there was a little bit of a trade-off there, but not massive differences. I think the bigger difference we saw was there are a few challenges of just kind of like naively applying this out of the box. If you just let it run, obviously those early diffusion steps are dominated by noise. So you start telling the model, hey, the first steps just do whatever. And that was pretty bad. So we ended up having to kind of hybridize, where the first few steps you kind of pretended you took the correct action, not in the actual trajectory you generated, but when you were then fine-tuning it on that trajectory, as well as doing some branching later on. There'd be some cases you looked like you were going to get a great pose out, and then the last step you just made a stupid little error, mainly due to the noise you added, like the step before. So we found if we added a kind of a bit of a branching process, we saw a bit of variance reduction. So we saw more impact from these kind of changes than from minor changes to the reward function.
Jim GSK here. I was wondering how your diffusion methods work if you have things like bridging waters, and because not all interactions are direct, and obviously that might also influence the affinity.
No, that's a great question. I mean, in terms of the methods we're using, if these things aren't taken into account by the underlying method, so for example, diffdoc doesn't see waters, we're not adding that in. We're just saying if we take the same baseline diffusion methods and train them slightly differently, how much do we see an improvement? So we're not addressing that fundamental representation limitation that you point out. But I think someone should.
Paul Emsley, Laboratory of Molecular Biology, Cambridge. For the challenges you mentioned towards the end of your presentation, do you have something like CASP in mind, or are you in a position to generate those ground truth crystal structures?
So I think the exact format of these kind of blind challenges is still in kind of discussion and exactly how it will run. I doubt it will be as intense as CASP, or it will be a very different format. But I think still kind of work in progress on exactly what that looks like.
Thanks for the lovely talk, Sophia Kadir from Evotech. Given the amount of data that you predict to include in the database in the next couple of years, how stringent will you be with ligand validation, given the new techniques that come up, so that we don't put more errors as such on your deep learning models? Thank you.
No, I think that's a great question, and I think there's always going to be some error there. I'm probably not the right person to ask about this as the computational person versus the experimental people. But I think already having trying to get paired data is a good start. Like if you have both an experimental structure and an affinity measurement, you can probably be a bit more confident that binding did occur. And then I guess if you have one or the other and you're kind of struggling, well, maybe there's more questions. It's not saying it doesn't bind, but there might be slightly lower confidence. And if you have neither, maybe it's still bound, but you didn't find it. But I guess you can have, again, a bit more confidence that probably not a binder. But I might look to direct you to some of my colleagues who are here at this meeting to talk a bit more about that.
Thank you. I think we're going to leave questions there.
Okay, I think we're going to leave questions there. There were a couple of more questions on the Slack, so if you can get to those, that would be great.